Hello everyone, and welcome to the second installment of these recorded lectures where we'll cover the rest of the content of this class on sexually transmitted diseases. You should have access to these slides on Blackboard as one intact set of slides, and in this segment we will cover HIV. And just as a reminder, we left off discussing the human papillomavirus, or the causative agent of venereal warts. And today we will talk about acquired immunodeficiency syndrome, or HIV/AIDS. So I'll bring those slides up now. AIDS is a recently characterized microbial pathogen, a viral pathogen of humans, first recognized and characterized in 1981. Since then, there's been quite a few individuals that have become deceased due to exposure to this virus. And you see that there's quite a few projected or estimated active infections of HIV globally. And that the distribution of those infections is not evenly distributed across the globe. There is a much higher concentration of infected HIV positive people in sub-Saharan Africa. And even in the US, there is a differential distribution of HIV positive cases within different segments of the US population as well. Now the rates of infection in the sort of more developed portions of the world have been relatively consistent to 1990, and this is mostly attributed to public health and education campaigns, teaching people about this disease, how it's transmitted, and how that disease transmission can be prevented or mitigated. There are two different classes of HIV, 1 and 2, that are differentiated based on their morbidity and mortality rates. HIV-1 is the more virulent type, and HIV-2 is less so. Both of them result in AIDS-like diseases. HIV-1 strains or group of viruses tend to lead to more rapid demise of the infected individuals. Now, we're all probably familiar with the routes of transmission of this virus. Similar to some of the other sexually transmitted diseases, be transmitted via direct contact between an infected individual and a naive host, and transmitted via bodily fluids to include blood and other semen and other bodily fluids exchanged during sexual contact. Many of the individuals that become HIV positive and progress through the different stages of infection don't necessarily die directly from the HIV infection, but due to secondary infections that are opportunistic in nature and oftentimes only found or observed in immunocompromised individuals. So, for example, there are pneumonia diseases that are caused by protists that rarely occur in otherwise healthy humans, but in those with HIV or severely depressed immune systems can become susceptible to this particular protist. And there are also folks that will die of cancers that are typically only observed in immunosuppressed individuals, things like Kaposi's sarcoma. Now, let's talk a little bit about the pathogenicity of this virus, how it recognizes host cells, and how it initiates its replication cycle. See on the top right-hand corner of the slide is an image electron micrograph of an HIV particle. You see on the surface there a variety of sort of donut-shaped or donuts on a stick, and those are all surface proteins that we'll talk about today that play a specific role in recognition of host cells and attachment, adherence, and introduction of virus into the host cell. Those specific proteins that we just mentioned on the surface of the HIV virus bind in a very specific way to a certain type of host cell, surface cell protein known as CD4. These are bound by a viral protein known as GP120, and the interaction of those two things results in fusion of the host cell membrane with the viral envelope and introduction of the viral capsid and the viral genome into the host cytoplasm as the initiation stage or the initial stages of infection. Here we see that same sort of interaction in a cartoon fashion. Again, this is an enveloped virus. On the surface of that envelope are these GP120 proteins that are virally encoded, and those will specifically recognize a CD4 protein on the surface of a host cell, initiating not only the surface recognition, attachment and adherence, and then also introduction into the host cell. This is a false colored electron micrograph showing sort of the outcome of that infection cycle, where you have a CD4 positive cell that is releasing newly replicated viral particles. So what are CD4 positive cells? Well, typically these are members of the cellular immune system in humans, specifically T lymphocytes that are responsible for the memory component of the cellular immune system. This binding of the virus to these CD4 positive cells results in a progressive decline in the number of viable CD4 positive T lymphocytes in the circulatory system, ultimately dropping that component or reducing that component of the cellular immune response down below sort of functional levels in the infected individual. So a progressive decline in functionality of the cellular immune response. There is a replication on the Blackboard site, or an animation on the Blackboard site sort of describes these steps in series, so you can watch that to get a feel for the process that's occurring here. When we look at the disease progression of this pathogen, you can see that plotted on this plot, and so what you see on the x-axis is time or months after HIV exposure. On the left-hand y-axis, that's the number of CD4 positive cells, of the concentration of those per milliliter or square millimeter cubic millimeter of blood, and that's the black line with the dots, and then the dashed line is represented by the number of HIV copies or genome copies, or a measure of HIV particles in the circulatory system. And what you see is that after the initial exposure days after exposure to HIV, the numbers of HIV particles are quite low, and then those increase over the first 1 to 2 months to being quite a bit higher, up to 10 to 100-fold higher than they would be at the time of infection. And that number can then dramatically decline within sort of 2 to 4 months post-exposure. And then after that time, there's sort of a sequential or very gradual increase in the number of circulating copies of HIV particles in the cell. This can make detecting HIV a bit more difficult, but not necessarily impossible with sort of very sensitive modern-day techniques. The other corollary you see happening here is expressed in the black line with the dots, the connected dots, sort of black line, is that post-exposure there is sort of a period of time up to a year and a half, during which the number of CD4 positive cells remains relatively constant in the exposed individual, and by about 18 months you start to see a significant decline accumulating in those CD4 positive cells, and then that process continues for the next anywhere from 3 to 10 or so years, where eventually the number of CD4 positive cells declines enough that opportunistic pathogens are able to establish and cause disease in an infected HIV positive individual, and ultimately those opportunistic infections and or a systemic immune deficiency results in death of the individual. And you see the corresponding increase in the number of viable circulating HIV particles in the circulatory system. So a progressive decline in the number of CD4 positive cells and a corresponding increase in the number of circulating viable HIV particles. So how is this disease diagnosed? Well, there's a couple of different tools. One class described there in the first 2 bullets deals with recognition of HIV-specific proteins, either on intact viral particles in serum samples or portions of those viral particles diagnosed or detected in a serum sample. These protein-based diagnostic tools can sometimes not be sensitive enough to detect the presence or provide a positive indication of HIV exposure in the first few days to a month and a half or so post-exposure, due to that time it takes for the number of viral particles in the circulatory system to increase. There are, however, DNA-based tools that can detect HIV particles at lower concentrations, either later on in the infection cycle or a little bit earlier in the infection cycle, using a reverse transcriptase or PCR-based tool or mechanism to amplify that viral signal in the sample from the potentially infected patient. There are different classes of treatments available on the market. Typically these are broken down into a couple of 4 different classes of drugs. Nucleoside analogs that act as competitive components that can be incorporated into a growing or viral genome as it's being replicated. As they get incorporated, they block additional replication of the viral genome, therefore blocking the replication cycle, the overall growth and replication cycle of the virus. There are non-nucleoside inhibitors that act as direct either allosteric regulators or competitive inhibitors or binders to a virus-specific protein that plays a role in viral genome replication. This is the reverse transcriptase, or abbreviated RT, specific to RNA-based viruses and not found, at least not found in normal host cell lines. There are also proteins that are therapeutics that bind to specific viral-specific proteases that play a role in cleaving viral proteins as they are transcribed in the host cell during a replication cycle and cleaved from an inactive to an active form. If you can block that cleavage step, you can block the viral replication cycle as those particles are being assembled in the cytoplasm. And there are also fusion inhibitors. And this class of therapeutics specifically targets the fusion proteins, or the specific conformations of the GP120 and CD4 proteins that play a role or become exposed upon binding of the viral particle to a susceptible host cell. So these are conformational changes in those surface proteins that are only exposed at the time of attachment and adherence of the viral particle to a susceptible cell line, and there are classes of either antibodies or other therapeutic proteins that recognize those specific or specialized conformational components or conformations of those GP120 and CD4 proteins. They can then bind those fusion components or fusion complexes and inhibit the invasion step, or the incorporation step that occurs after the viral particle recognizes a CD4 positive cell. This is a longer list of the specific therapeutic agents that are available. And again, they are broken down by the classes that we just discussed. So I won't ask you to memorize all those different drugs, but rather know the different classes and mechanisms of activity of those classes of HIV therapeutic agents. This last slide is a summation of different strategies for targeting pathogens. For both, this could be used for humans or animals or plants or what have you. This does not deal with viruses per se, but the strategies that are being used here could be applied to viruses. And specifically, the strategy that's being applied and sort of summarized in the slide is that there are different classes of therapeutic agents that target specific processes, compounds, enzymes, or structures that are found in pathogens and not found in the host cell lines or the host tissue. So, for example, in a prokaryotic pathogen, you could target cell wall synthesis because the prokaryotic pathogen has a peptidoglycan cell wall, and the host cells, at least in animal host cells, there is no cell wall, nor is there any peptidoglycan-like structures that use similar enzymes or pathways to be created. Therefore, if you target that prokaryotic specific process, you can specifically target the pathogen without harming the host. Same kind of rationale can be applied to protein synthesis. Again, this is synthesis of processes that also occur in eukaryotes, but targeting the structures that are specific to the prokaryotic ribosome. You can also target ribosomal processes for eukaryotic pathogens, but the application of those therapeutics has to be done in a balanced and appropriate way so that you're not applying a high enough concentration of a given protein synthesis inhibitor that targets a eukaryotic pathogen and using that in a eukaryotic host, because then the eukaryotic host would also have its protein synthesis processes blocked, and then that therapeutic agent could be toxic to the host. So sometimes that's a timing issue, a location issue, or site of application issue, or a concentration issue, so that you can adequately target a eukaryotic pathogen without harming the host. Again, that strategy can be used for viruses. We just mentioned a few examples for HIV. Same things apply for all the other viruses that are pathogens of, say, humans. If you want an effective therapeutic, you need to target a structure or process that's unique to the pathogen and not present in the host. So what I'll ask you to do here is Not necessarily memorize all of these antibiotics that are listed under these different classes of things, but do know the different strategies or targets or classes of targets that are represented here, like folic acid metabolism, cell wall synthesis, DNA gyrase, and so on, and know a couple of the examples under those categories as they apply to specific pathogens that we've discussed already. So we've mentioned a couple of prokaryotic pathogens already, for which quinolone antibiotics or inhibitors of DNA gyrase are used as therapeutic agents. What you want to know is remember or review what DNA gyrase does in prokaryotic organisms, and then link these specific quinolone antibiotics to the diseases that we've talked about already. That should narrow that, or that will narrow the list of antibiotics that are presented on this slide. But again, remember, or be sure to learn all of the different classes that are here, and then memorize or learn those example antibiotics or therapeutic agents that are used for the pathogens that we've talked about already, and or we will talk about in the next segment of recorded lectures. That's where we'll stop on this segment of STDs, so I encourage you to review this material, and then keep an eye on the Blackboard site for other recorded lectures associated with the rest of the human microbe interaction content that we'll talk about this week. So I'll just switch out of this presentation and stop the recording